Welcome friends. Today we will be discussing about these various sampling strategies which are involved in collection of the data so that a predictive analysis about the qualities and parameters of the formulation can be done. So there are different strategies and different methods of analyzing this data which are being utilized and have been prescribed by the regulatory bodies as a mandatory requirement for filing as well as for analysis of the quality of the products. So we will be looking into few of these aspects in this current lecture. So in this lecture we will be looking into the principles and importance of pharmaceutical sampling, the binomial probability distribution and interpreting the operating characteristics for decision making in lot acceptance and rejection, evaluating the relationship between defect rates and probability of accepting or rejecting lots under statistical models, how calculation of the required sample size and detecting meaningful differences can be done. Also interpreting and using the Z values for hypothesis testing and power analysis is done. Also assessing the statistical power of a test and determining its ability to detect real differences where they exist in the dosage forms can be beneficial. So sampling is the process of selecting a certain number of items from a population to make conclusions about the whole group. So a sample is generally a representation of the complete population characteristics. In pharmaceuticals, sampling helps ensure product quality by checking samples of drugs, raw materials, intermediates and the final products. When the sampling occurs, sampling is done either before, during or after the manufacturing and packaging operations. Quality control inspectors must have the authority to sample them at any stage of the process. This ensures the product meets the quality standard and supports the quality assurance program. The sampling requirements must follow proper techniques, requires an adequate number and size of samples, and the entire batch is judged based on the sample taken, making accuracy very critical, because this can result either in the rejection of the complete batch, which may be a non-defective batch, or acceptance of a defective batch. So the need for testing is there because despite control systems, final dosage forms must still be tested to confirm quality, because there may be errors because of manual handling, machine errors. Also, the variability in process steps affects the uniformity of components in the dosage form. The sampling thus has an impact on the accuracy of the sample, directly affecting the reliability of the results. Improper sampling can introduce bias and error. Well designed plans reduce sampling errors. 100% inspection is ideal. It is impractical and may also introduce human errors along with fatigue. Good sampling is preferred over 100% inspection for efficiency and accuracy. The statistical sampling plans should accept high quality batches and reject substandard ones efficiently. The stringency of the plan determines how well unacceptable units are controlled. The standard sampling plans are prescribed by the government sponsored agencies. Common examples are the MIL STD 414 for variables sampling plan, and the other one 105D is for attribute sampling plan. The advantages of sampling are that it saves times in designing sampling procedures. They are widely accepted by industries across the globe. The contents of the standards provide sampling procedures and reference tables. These are used for planning and conducting inspections. Now, the batch acceptability is determined based on the acceptable quality level linked with the sampling plan. The plan defines the number of units to inspect, that is the sample size, the criteria for batch acceptance or rejection, inspection level. This defines the relationship between the batch size and the sample size and must be set by the responsible authority. The common sampling methods could be single sampling, fixed sample size can be inspected. Decision to accept or reject the batch is made after this single inspection. This uses one acceptance number, maximum allowed defectives, for the acceptance or rejection of the batch. There could be double sampling. If the first sample result is inconclusive, a second sample is taken. Two acceptance numbers are used, one for the first sample alone and one for the combined total of the defectives in both the samples. Triple and multiple samplings can be done. These are extensions to the double sampling. This involves sequential inspections with added flexibility for decision making. This is used to potentially reduce the average sample size inspected. For example, in the initial sample, the size is of 50 tablets. So, the decision is based on the first sample wherein less than or equal to 2 defectives are there. So, the batch would be accepted. If more than 4 or equal to 4 defectives are there, the batch would be rejected. If 3 defectives are there, then second sampling needs to be done. So, second sample size will be again 50 tablets and final decision is based on the combined samples. So, if the total defects in both these samples is less than 4, the batch is accepted, and if the total defectives is more than or equal to 4, the batch is rejected. The construction of a statistical sampling plan normally requires that 4 basic quality standards be specified. In general, the sampling scheme should be designed in such a way that the alpha and beta errors are appropriately shared by the producer and the consumer. So, an acceptable quality level, a batch of tablets is considered to be accepted if it contains 2% or less unacceptable tablets. Then an unacceptable quality level is there. Example, the same batch of tablets is said to be rejected if it contains 4% or more unacceptable tablets. The risk or error designated as alpha, that is the producer's risk, which is the probability of rejecting a good batch, and the risk or error designated as beta, which is the consumer's risk, is the probability of accepting a bad batch. So, the key parameters in the sampling plan are the acceptable quality level, which is the maximum percentages of the defectives which are acceptable, unacceptable quality level wherein minimum percentage of defectives are considered unacceptable. Risk of rejecting a good batch is alpha and risk of accepting a bad batch is beta. If the AQL and UQL are close and beta and alpha are small, large sample size is needed. However, if the AQL and UQL are far and alpha and beta are large, sample size may be small. The operating characteristic curve is a graphical tool to visualize the performance of the sampling plan. So, plots of percentage of batches expected to be accepted is on the y-axis, whereas percentage of the defective in the submitted batches is on the x-axis. This shows how well the sampling plan distinguishes between acceptable from the unacceptable batches. Every sampling plan has a corresponding operating characteristic curve. It is a graphical tool used in sampling plans to show how likely a batch will be accepted based on its quality. The horizontal axis shows the percentage of defective units in a batch and represents the actual quality of the lot. Vertical axis shows the probability of accepting a batch with a certain level of defects from 0 to 100%. Indicates how often such batches pass inspection under the sampling plan. So, the key terms in the curve are, that is AQL, which is percentage defects considered unacceptable, unacceptable quality level, percentage defects considered unacceptable, alpha choosing of chance of rejecting a good quality batch, and beta, which is chance of accepting a poor quality batch. And n is the sample size, that is the number of units taken from the lot for inspection. Now, the example wherein the batch size is of that is the number of samples taken is 10. So, if a batch has 4% defectives, that is AQL is 4%, there is a 90% chance then that it will be accepted. Alpha is equal to 10%. If a batch has 20% defectives, that is UQL is 20%, there is a 20% chance that it will be rejected. Beta is 20%. So, different curves represent different sample sizes. The larger the sample size, the more accurate is the decision making. For example, or a sample size of 10, AQL, if it is 1%, the acceptable defect rate, batch is accepted 90% of the time, alpha is 10%, that is risk of rejecting a good batch. UQL, if it is 20%, that is unacceptable batch defect, is accepted 5% of the time, that is beta is equal to 5% risk of accepting a bad batch. Increasing the sample size reduces the tolerance for defectives in the batch. With larger samples, the sample becomes more discriminating, better at accepting good and rejecting at bad batches. At the same AQL, large sample size improves inspection accuracy and reduces both producers and consumers' risk. The weight variation and the OC curves are developed to evaluate how likely a batch is to pass the compendial weight variation test. Examples with 5% defectives. A batch with 5% underweight or overweight tablets had a 93 acceptance probability when 10 tablets were sampled. As sample size increases, the acceptance probability drops for bad quality batches. For a batch with 20% defectives, probability of passing is 40% with 10 tablets, 23% with 20 tablets, 4% with 50 tablets and 0% with 100 tablets. So, tablet batches often consider 1 million units treated as infinite population statistically. For such large batches, the large sample sizes up to 200 units are required for the reliable reporting. According to MIL STD 414 variable sampling plan, larger sample sizes are recommended for high accuracy evaluation in the large batches. The dot sampling plan is continuous, not batch based planning. Initially requires 100% inspection until a defined number, that is P, of consecutive batches of tablets are defect free. Once you get defect free samples, after meeting the P value, for example 50 defect free tablets if I have got in a row without break, the sampling then shifts to the fractional method wherein randomly samples are inspected of the tablets. So, then again it switches to full inspection if the defective tablet is found during the fractional sampling. 100% testing is resumed until another set of P defect free tablets are achieved. So, on a rotary tableting press with 25 punches, a batch of making a batch of 1 million tablets. So, P is 50, that is 2 tablets per punch. So, F is 1 per 10,000 tablets, that is upon testing. So, an additional then 100 tablets might be tested if no defects are found and process runs smoothly. This reduces the testing workload when the process is operating as expected, increases chances of detecting issues when tablets fall out of specification. So, a sequential sampling plan helps, it goes from the continuous testing, then that is full inspection, to fractional, and then once defects are obtained, then again it switches to full inspection. Now, the emphasis is placed on ensuring sample homogeneity for accurate statistical evaluation. Oral and parenteral solutions are typically homogeneous. For such forms, sample sizes as small as 1 unit are often sufficient. Similarly, a homogeneous drum of powder, drug or excipient can be treated like a solution. Single or duplicate samples are usually adequate. For forms like ointment, creams and suspensions, these are treated like solutions if assumed homogeneous. However, multiple samples may be needed to verify homogeneity. If heterogeneity is suspected or known, the batch must be treated as having an infinite population of variable components for sampling and statistical purposes. So, the sampling in statistics is important, and the importance and basics of statistical sampling is important, as samples are small groups of observations taken from a larger population. The goal is to estimate population parameters, measurable characteristics using the sample. Inference from samples to population is a core function of statistics. A clearly defined population is essential to ensure the sample is representative. So, 100% sampling is not feasible due to time and cost constraints. Destructive nature of the sampling, inaccessible populations, for example, all patients in a disease cannot be accessed, and good experimental design depends heavily on proper sample selection. So, there are various kinds of sampling methods. This can be categorized into probability sampling. preferred for statistical validity, and non-probability sampling, also called as authoritative sampling. There are different types of probability sampling, that is random sampling, wherein each member has an equal chance of being selected. Stratified sampling, the entire population is divided into strata and samples are taken from each stratum. Systematic sampling, every nth item is selected for sampling from a list or sequence. And cluster sampling, entire group or clusters are randomly selected rather than the individuals. So, random sampling is generally used in pharmaceuticals. Each experimental unit has an equal chance of selection. This produces unbiased fair estimates of population parameters. In tablet sampling, each tablet must have an equal chance of being chosen for assay and visual inspection. Now, the methods which are used to ensure randomness are numbers are assigned to all the units and they are selected using random number tables. Thoroughly mixing all the units physically or metaphorically and then drawing like a lottery. Now, bias can be unintentionally introduced if randomization is not carefully implemented. In clinical trials, full randomness may be impractical due to logistic issues. Thousands of clinical sites are there. Convenience sampling may be used, but stratifying by important factors like geography can simulate randomness. Alternative approaches use stratified sampling across different time points during production, and pseudo-random methods can be creatively designed to mimic randomness in challenging situations. In stratified sampling, the population is divided into distinct subgroups, that is strata, and random samples are taken from each stratum. Strata differ significantly from each other. Items within a stratum are homogeneous. So, these are the two major criteria which have to be followed in stratified sampling. The advantages of this type of sampling are that they reduce sampling error when within stratum variability is low compared to between stratum variability. In pharmaceuticals, sampling of 10 tablets every hour during a tablet production run can be kind of stratified sampling. This is useful in quality control for targeted representative sampling across time or other stratification variables. Then there is systemic sampling. The samples are selected using a fixed interval, for example, every nth unit or every nth time point. Testing every 100 tablet produced on a manufacturing line can be an example. This ensures consistent coverage across a process, often more practical than simple random sampling. However, if the process is cyclical and the sampling interval aligns with the cycle, it may produce biased or misleading results. Cluster sampling is dividing the population into clusters, each containing multiple subunits. A random sample of clusters is selected and subsampling is done within each cluster. For example, in a batch of 10,000 bottles, each with 100 tablets, 100 bottles are selected randomly, that is clusters, and then 10 tablets per bottle, that is subunits, are inspected. This is useful when population is naturally grouped, an example package unit. This ensures cost effective and practical approach large scale inspections. Now, the goal of sampling is to ensure quality and complete representation of the entire bulk material. A representative sample should thus reflect the true characteristics of the bulk if it were homogeneous. Improper sampling only from the top of a container or bottom can lead to biased results. No strict sampling methods are prescribed or they exist. These often rely on empirical rules and experience. For bulk raw materials, sample size often is based on N containers. Using a sampling sheaf to collect material from various container levels is done. The use of sampling sheaf to collect material from various container levels, then combining and thoroughly mixing samples to achieve uniformity is done to ensure uniformity as well as homogeneity before analysis. Properly executed sampling leads to accurate quality assessments of raw and finished products. Many data analysis assume independent sampling points. Each observation does not affect others. Independent data ensures valid statistical reference. However, dependent or correlated observations can also occur when Repeated measures are done on the same individual, for example, blood pressure over time, repeated assays for the same portion of material, or multiple responses from the same individual, that is taste tests. So, data may appear independent in one context but may be correlated in another. Duplicate assays on single bottle over time are likely not independent if performed under same conditions, same analyst and reagents and equipments. More independent if done in different labs or different times with different conditions. So, they are more independent. Using tablets from one bottle introduces homogeneity, which reduces independence compared to using tablets from across the batch. Correlated observations require special statistical analysis to account for the dependence. Now, bias can easily enter if sampling not done carefully. Human subjectivity often introduces unintentional bias. Randomization and blinding are key tools to reduce the bias. Randomization avoids selection bias. Blinding ensures objectivity during evaluation, especially with subjective outcomes. Examples of bias are like selecting more severe patients for the treatment is believed to be better, using first assay results to judge second assay outcomes, rejecting data that does not fit expected trends in the curve fitting methods. Now, sampling for attributes and operating characteristics curve. So, the binomial distribution is useful in constructing acceptance rejection of the sampling plans and quality control. The MILSTD 105D is a common reference that outlines the attribute based sampling plans. These plans specify the number of items to be inspected, the number of defects allowed before rejecting the lot. The sampling aims to balance inspection rigor based on seriousness of defects and risks of incorrect decisions, accepting a bad lot, that is type 2 error, and rejecting a good lot, which is a type 1 error. So, for example, there is a plan designed for lots with 0.25% defect rate, that is AQL is 0.25. In the sampling plan, 500 samples are randomly selected. If 3 or fewer defects are found, lot is accepted. If 4 or more defects are found, the lot is rejected. The probability of acceptance when defect rate is 0.25% is equal to the binomial probability of observing 0, 1, 2 or 3 defects in 500 items. So, this is how the binomial distribution curve is, and the operating characteristic curve for the plan N. The probability of accepting a bad quality lot is a critical concern in a sampling plan. The OC curve illustrates the probability of accepting a lot based on the true percentage of defects in that lot. The probability of rejecting a lot is calculated as probability of rejection is equal to 1 minus probability of acceptance. To construct an OC curve, compute the probability of acceptance for various assumed percent defective values, that is 0.25, 0.5, 1%, etc. Then important points are based on explanation of acceptance and rejection probability in the sampling. The probability of a non-defective item is given by equation Q is equal to 1 minus P, which is equal to 0.99, and for plan N the probability of accepting a lot with 1% defect is 0.26%. Therefore, the probability of rejecting the same lot is 0.74%. Interpretation: a lot containing 1% chip tablets will be rejected about 74% of the time and accepted 26% of the time using the sampling plan. This highlights the risk of accepting bad lots and the importance of choosing the appropriate sampling criteria. Now, the sample size is important to be estimated because this is crucial to detect a statistically significant difference if a true difference exists in the samples. Now, using this equation, which is mentioned on the right side, wherein D is equal to difference to be detected, alpha probability of type 1 error, beta probability of type 2 error, which is false negative, N is the sample size. Again, if any three of the given parameters are there, the fourth can be determined. To compute sample size N, we must specify variance of the data, alpha, which is the risk of wrongly rejecting the null hypothesis, beta the risk of wrongly accepting the null hypothesis, that is type 2 error, and D the minimum difference considered meaningful. So setting appropriate values for these parameters is often required, which requires experience and judgment, and the choice of alpha and beta values affects the power and reliability of the study. So the Z values are used in sample size calculations and are the same as those used in the hypothesis testing. So 1.96 for a 5% level of significance, which is alpha is equal to 0.05, and 2.58 for a 1% level of significance when alpha is 0.01. Z beta represents the Z value above which the beta probability lies in the upper tail of the normal distribution. The common Z beta values have been given. So the problem involves determining the sample size necessary to detect meaningful differences in the tablet weight. A difference of 3 mg or more from the target tablet weight is considered important to detect. If such a difference truly exists, there should be a 90% chance, that is power is equal to 0.9 or beta is 0.1, that a statistical test will detect it. The type 1 error rate is set at 5%, meaning there is a 5% chance of wrongly concluding a difference exists when it does not. So the specified difference 3 mg and the alpha beta risk are not fixed standards but are chosen based on judgment, experience and context. These values are typically ballpark estimates unless legal or regulatory requirements demand precise definitions. The process highlights the importance of careful planning in designing these experiments. If the standard deviation is 10, n is calculated accordingly. A statistical test comparing the observed mean weight versus the target weight would be then where having alpha and beta risk of 0.05 and 0.1 respectively. The values 1.96 and 1.28 in the calculations of n refer to the standard normal deviate for alpha at 0.05 and beta, which is 0.1. Then comes the power of a test, which refers to the ability to detect a true difference when one really exists. Power is mathematically expressed as 1 minus beta, where beta is the risk of type 2 error failing to reject a false null hypothesis. So to conclude, the pharmaceutical sampling is a vital part of quality assurance, ensuring that defective products are identified and rejected before reaching the consumers. The statistical tools like binomial distribution, OC curves and sample size calculations enable informed decisions that balance quality, assurance and efficiency. Understanding the alpha and beta errors, acceptable defect limits and power analysis empowers researchers and regulators to design sampling plans that are both scientifically robust and practically relevant. With careful planning and statistical insight, one can ensure product safety, regulatory compliance and optimal resource use in pharmaceutical manufacturing. So, thank you very much for your patient hearing, and I hope you would have understood why sampling is important and how it helps in accepting a good batch and not rejecting the good one. Thank you very much.